right. Trying to cut down my coffee. See if that helps.
Oh my God.
I'm like, screwed up. This is me sober. Like, sobered up. The last 6 months and now I'm like, scary kid ever. You guys are so quiet. So I'll reward you though for coming today. Remind me, stop me if I go too far. Just an FYI, this exam, the next lecture exam is scheduled for 2 weeks from today. So I anticipate, so most of that exam is going to be multiple choice questions because it's a relatively large class. I know it doesn't look like it today, but most of these seats are filled whenever it comes to exam day. So for that reason, just for the grading purposes, the other thing too is I know what it's like to be an undergrad. I remember like how you set weight for a professor to hand back an exam, the anticipation of what that will be. So I try to return, if you take an exam on Friday, you try to return them on Monday, and by the latest on Wednesday. They went horribly wrong, so like scanning the, and then also, I also bring the scan-a-thons, so you just have to bring a pencil and yourself. Yeah, bring your brain for sure. And ask that you follow specific rules, especially rules of engagement in the classroom when you're taking the exam, that you don't have any material out and all your cell phones and implements of destruction, potential destruction are put away. So, are there any questions about material that was covered in chapter 2? I know I went through some of that really fast, but a lot of it's really not conceptual information. A lot of it is just like historical context and historical basis. And it's all material that's going to be, I talked about the pattern of evolution. It's certainly fair game for the purposes of the exam, but it's all information that's going to be covered in detail in subsequent chapters for the most part. It's not something where I'm just throwing you out of it. Okay? So yeah, let's go ahead and get started. Let's do it. You read my gatekeeper today, Simon? Will you stop me? 5 minutes, at least 10 minutes before the end, that way we can talk about potential exam questions. Okay? So, and then if I ask you what time it is, well, I don't know. Anyway, just to remind you. Yeah, the time should show up. Okay. I want to go back just on chapter 2 and just finish up that last slide. I kind of wanted to do it at the end of last lecture, but at the same time, I think it's more appropriate to sort of summarize information. Fully plated. Okay. All right. So essentially, with the information that's been accumulating over a number of years by a lot of different scientists who are involved in looking at the experimental approaches to evolution, but scientists who are also involved in paleontology. So they have they have a really good understanding of the fossil record. And, information that's compiled across all these different disciplines. We're talking about vertebrate anatomy, we're talking about from a zoologist to botanist. All of these different disciplines are sort of combined, and we're looking at the mechanism of evolution well, the mechanism of evolution as specifically outlined in Darwin, but we're also looking at a broader pattern too that encompasses speciation, which we will later come back to in terms of macroevolution. So here's what we know at this point, is that really we're most likely, the biological diversity that we generated in terms of the Earth, what we were looking at, unless God had a particular fondness for beetles, stars, okay, was not all divided and created. We know now that there's a lot of change in terms, well, there's a lot of change. It's going to be very rapid change, and sometimes it's going to be very long-term changes. That's what we see to generate all this biological diversity that we find. So, we also find that lineages, in this case, in special creation, that we find that the species change over time. We find new lineages split and diverge. I'll introduce a lot of that, especially when I talk about chapter 4, an introduction to phylogeny and the process of speciation, is that we find these changes over time. New life forms are generally derived from older forms. We're going to talk about that, especially when we talk about chapter 16 and subsequent chapters. When we talk about speciation and extinction, I presented some information that all life forms are related by common ancestry. We haven't talked necessarily in detail about that. Do you guys know what the 3 domains are of life, though? You know that now we don't just divide organisms into kingdoms, but we divide them first into 3 domains. Any idea what those are? You really can help me out? Shout out some ideas.
Archaea.
Archaea, that's good. That's one domain. Archaea were traditionally before classified as bacteria, okay? So they look a lot like bacteria. I mean, they basically function like bacteria, but a lot of them are able to use different substrates that bacteria have not been used, or they can't utilize, especially methane, hydrogen sulfide, things like that. So we find them, kind of rapid discovery of them in deep sea hydrothermal vents and other geological methane producing pools. Another one. What?
Animalia.
Yeah, so Animalia, so animals, plants, fungi, algae, you name it, are in one domain. And that one domain is something that we call the eukarya. Okay? So we have archaea and eukarya. So the eukarya is anything that is considered a eukaryote. Okay, so these are all the organisms that we have that have organelles, specified organelles, especially mitochondria, and in some cases, such as plants, chloroplasts, right? So they utilize specialized substructures for cellular respiration and obtaining a carbon source. So they're all classified and all lumped into one domain called eukarya. So there's one other domain, I heard it mentioned earlier, bacteria, right? So we have archaea now, bacteria, and eukarya, okay? So we know that they're all related, and there's the descent in this case in common ancestry, but it's a long-term evolution. We're talking about millions of years since the archaea, for example, and the eukarya diverged. Okay, so we'll come back and talk about that all in detail. Do they cover domains in 1120? Yeah, they do. Or 1110? Yeah, I didn't know if it was 1110. They started it in 1110. Okay. All right. Now we're going to talk about chapter 3, one of my favorite chapters. Mechanism of evolution by natural selection. We're going to finally cover this in a lot of detail. We're going to talk about adaptation. We're going to talk about traits. We're going to talk about phenotypes. We're going to talk about genotypes. Okay. This is what we're going to cover. We're going to cover mechanisms of evolutionary change. I'm going to introduce you to all the mechanisms that result in evolutionary change, which is defined as a gene frequency. Finish that for me. Over time. Okay. It can be a short period of time, and it can be a long period of time. Right? Short period of time. People were generally surprised that it could be so short. I'm sure Darwin would be be surprised at some of the data that's shown that natural selection can operate in very short evolutionary periods and even over the course of a single one generation period. We're also going to talk about the difference between fitness and adaptation. We're going to outline, in this case, Darwin's 4 postulates. And I'm going to illustrate with some very important and influential experiments that have been done the mechanism of evolution by natural selection. These people have really kind of like set the forefront, they've set the bar really high in terms of outlining and testing these 4 postulates. And so I want to illustrate some of their work and some of the important discoveries. And then we're going to talk about the nature of natural selection. Okay, so as I mentioned before, and this is going to be important, it'll be important for the purposes of the exam to understand. Evolution, in this case, evolution occurs by multiple mechanisms. Evolution isn't just covered under a single mechanism. I would say that natural selection is a very important mechanism, given how it's been illustrated in terms of our biological diversity. We've accumulated a lot of ample evidence that natural selection is a very important mechanism. It doesn't operate uniformly in all species, okay? But under certain contexts, many different species have evolved because of the mechanism by natural selection. So that's what we're going to be focused on today. I also mentioned before, or maybe I mentioned, I teach another class in evolution called Analyses of Genetic Markers. So sometimes I'm like, I mentioned this before, but really I talked about it in that class. So you'll have to correct me if I'm wrong. Have I talked about genetic drift in here? I at least mentioned that the mechanism of evolution, right? So you probably were you should have been introduced to this in 1110, the concept of genetic drift, where they talk about 2 mechanisms of genetic drift. One is called the bottleneck effect, and one is called the founder effect. Okay, so those mechanisms, which are that operate in small, and those mechanisms can alter gene frequencies. So you can have a change in gene frequency over time due to catastrophic loss of individuals, which results in a bottleneck, okay? Or having a few individuals that found a new population. By random chance alone, those few individuals may not have the same allele frequencies as the original population. So rapidly, you can have a change in gene frequency over time. So I think there are 2 very important mechanisms. And if I were to number these, number 1 would be natural selection, and number 2 would be genetic drift, okay? And genetic drift is very important, especially if you work with organisms that, like I do, that have airborne spores, that are colonizing new populations or colonizing new geographic areas where only a few of the individuals from the original population are colonizing, okay? So, now, these are 4 forces of evolutionary change. So when I say the mechanisms that drive a change in gene frequency over time, most commonly result from natural selection, and they most commonly result, in this case, from genetic drift. Now, the only way, though, that you can introduce new alleles, new genetic variation in a population is through the process of mutation. That's one way to introduce new things, is to have mutation. How does mutation happen? Mutation happens because sometimes DNA replication is a little bit sloppy, okay? So it happens when our cells undergo mitosis, or when our cells undergo meiosis. We can introduce new mutations. Our cells, in certain parts of our body, are undergoing mitosis at a very fast rate, okay? So there is a possibility of introducing a new mutation. Now, mutations aren't necessarily a mechanism of evolution. They are, okay? They result in the change in the DNA. That change in the DNA is the raw material for evolution to occur. It needs that change, yet for evolution to occur, but it's not evolution in the process, okay? Once you have that genetic variation, then you can have natural selection or genetic drift. Gene flow is not necessarily as well a mechanism of evolution, but it can cause a change in gene frequency over time. Just by individuals with different allele frequencies migrating to a population, you can change the composition of that population. So it's not necessarily a mechanism. Mechanism is something like natural selection and genetic drift, which we are going to spend more time on than either of these two principles that are operating in a population. Okay, so back to Darwin. When Darwin outlined his mechanism of evolution by natural selection in The Origin of Species, he outlined the concept of an adaptive trait, okay? And so we're going to talk about what an adaptive trait is. So he basically said individuals that have adaptive traits are more likely to have a higher fitness. When I say the word fitness, there's two words that should come to mind that should just be starting to be ingrained in your head when I'm going to talk about fitness the whole semester. Survival, okay? So individuals have a greater survival and they have a greater reproduction. So they have a higher likelihood, in this case, with higher survival and reproduction, of passing those genes off to future generations. Now, fitness is also going to be relative. In a sense, it's really hard to, you can't just say, oh, that's an adaptive trait, okay? Basically, what you have to say is individuals that have a higher survivorship, okay, survival, higher survivorship and reproduction, that have these specific traits that lend higher survival and reproduction relative to other traits in the population is an adaptive trait. Okay? So when people say, oh, yeah, I understand, you know, I don't believe in evolution, but I believe in adaptation. And then I ask them what their definition is of adaptation, and their adaptation is any trait that is found in a future generation. But the way that evolutionary biologists then define an adaptive trait is a trait that allows greater survivorship and reproduction relative to a trait that doesn't provide greater survivorship and reproduction. The other thing that people like to say to me, too, about evolution is like, oh, well, the strong evolution by the mechanism of evolution means that the strongest survive. As if strong, in this case, imparts some relative fitness. Now it may, in some sense, certainly probably impart higher survivorship and or reproduction, especially for rock climbers. Since I don't have individuals who are generally tend to be very strong, who generally on big walls and rocks have higher survivorship and therefore reproduction. It doesn't mean that the strongest survive. All it means, though, in the context that we are talking about it, is individuals that have adaptive traits survive. But remember this, what is adaptive is also relative, because the environment is constantly changing, right? So a trait may be adaptive in one environment and be non-adaptive in another environment. So in this case, a trait can vary depending on the environment that the individual resides in, or that the individual lives in. Okay, so how, in a sense, do adaptations arise? Well, one mechanism is the evolution by natural selection. So Darwin outlined these four postulates, and you have had an introduction, at least, a very brief introduction to these four postulates when we talked about the evolution, in this case, of HIV to AZT. We also talked about it in the evolution, we talked about it in the evolution by the mechanism of natural selection of bacteria based on antibiotic resistance, right? So that's where we outline these 4 postulates. So I'll go up there briefly. Individuals in a population differ from one another. How do we define this difference? Variation, right? Variation, though, is defined really at the genetic level, yeah, at DNA, okay? So I look out and I see variation in my classroom, okay? I see variation in terms of traits, but I know that that variation in this case is going to have a genetic basis. So I see variation in terms of diversity, but I know that that diversity has a genetic basis. So remember, this is the raw material. You guys are fuel then, in essence, for evolution. We couldn't have evolution in this case if we didn't have multiple individuals where there was no variation. So you should embrace diversity. You should embrace genetic variation in a population. These differences that we see are at least in part passed from parents to offspring. This is also something that we call heritability, right? Remember that? I introduced the concept of heritability. We know that has to be a genetic basis. Darwin didn't know what it was. He didn't understand really that DNA was in cells and that not in part, but in full, that it created all this stored genetic basis for future generations. He knew there was something there, he just didn't know what it was in terms of the molecule. Now, some individuals are more successful at surviving or reproducing than others. Remember that I defined this as fitness. So what we're saying is, some individuals in this case, individuals in a population are going to experience competition. There's going to be a limitation of resources. Where did Darwin get this idea? Does anybody remember? Yeah, Thomas Malthus. Remember that he wrote very important paper in Principles of Economics, outlining that there was most likely that resources were not unlimited. Okay, that there was a limitation to resources, and essentially there was going to be a competition between resources. We know that to be true. Much more true in this case in natural populations, which I like to study rather than human populations. I think human populations also have a limitation to resources. They just haven't necessarily, at least in our context, discovered or what those limitations are going to be. Okay, what kind of crisis is going to develop because of the fact that we use so much resources. Number 4, though, the differences in fitness. Remember, differences in fitness. Fitness is going to be described as survivorship and reproduction or non-random. What do I mean by non-random? There's something out external that's influencing that, some environmental stimuli usually that's influencing that. That means, too, that some individuals are going to increase with the frequency and other individuals are going to decrease with the frequency. It's not random with respect to which individuals, okay? But individuals with an adaptive trait are going to increase with the frequency of that particular trait in the future generation. And individuals that lack that adaptive trait are going to decrease in the frequency of in that population. So, what do we have? Fluctuation. Fluctuation in genes over time. Okay? What do we have, really? That evolution is operating at the level of the individual, but the consequences on the population. And that's why when I look out at you guys, for example, I say evolution cannot occur. Okay? Unless we have the raw source of genetic variation, which you see with the number of individuals, okay? And that those differences in this case are going to be non-random. Some of you are going to have traits that are adaptive, some of you are going to have traits that are not adaptive. And it's not going to be random with respect to which individual we pull out of the population. It's going to depend on the environmental context. Okay. I want to talk about one of the most influential experiments in terms of natural selection. And that is like looking at the evolution of poop color. This was a really significant study because First of all, they were able to test all the postulates. I'm not able to cover every single paper that was published in a lot of significant journals, big journals, but I'm going to cover or highlight some of the important research and show how it was used to test each of the 4 postulates. So first I want to introduce you to the study system. And you might be like, mice, that's boring, whatever. Okay? But it's actually not boring from an evolutionary context. What's interesting about this is that you basically have this population up here. Oh, here it is. Okay. This population up here, and these are referred to as old-field mice or mainland mouse. Okay, so they're found in the southeastern United States. They occupy the great states of Florida. In this case, this is the great state of Georgia, Alabama, and all the way up into South Carolina. Okay? So, these mainland mice are only typically found, well, they're not completely restricted, but the majority of them that have this really dark colored coat are found in what is referred to as old field, which is really former agricultural sites that these mice have invaded. A very dark and a very loamy soil, though, in these agricultural fields, these abandoned agricultural fields that kind of have grown up with a lot more weeds and things like that. But the soil in this case, in these areas, is really dark. Now, interestingly enough, though, the same population now this population has actually invaded many of these kind of sandy coastal dunes of, well, these 4 states, but we're going to talk about them. These sandy coastal dunes that are primarily the populations that we're going to study are found in Alabama, Georgia, as well as parts of Florida along the Atlantic coast. Interestingly enough, can you guys see this phenotype change though? Most of these sandy beach mice, they're all considered different subspecies, these populations outlined. This is along the Atlantic coast, and this is along the Gulf Coast. All of these are considered different subspecies. But there's no reproductive isolation, meaning they're not different species. They can still reproduce together. It's just that you can look at them and find some morphological distinctions. The biggest morphological distinction, the most obvious, is what? You guys? If you go from here to here, from here to here, what do you see? Change in coat color, right? So these are found in dark soil. These are these beach mice that are only found in white sand beaches. And we've seen a shift, right, in coat color. Individuals that occupy these old plantations in darker colored soils are brown, darker brown. Individuals that occupy only these sandy beach dune habitats or other islands are all lighter colored. They're more closely related to white. Or more closely, they're higher frequency of light colors. Okay. So, what I'm not going to introduce today is a lot of the genotype, but I'm going to talk specifically about the phenotype in this case. Really, what we're talking about is the inheritance, in this case, of coat color. One other thing I want to mention, and this isn't going to be important for the purposes of the exam, but it's going to be important to illustrate that this can happen over relatively short time scales, is that it's thought, too, that all of these mice with this lighter coat color that occur on these sandy beaches, most of those sandy beaches were created geologically within the last 7,000 years. And it's thought that really the colonization by the old field or the agricultural mice with the dark color happened, okay, in this case, in some cases independently, but one ancestral population of mice likely colonized this. We've seen these significant coat color changes within the last few thousand years. Okay, so these are relatively young, geologically, compared to what we're used to studying. All right, so one postulate, the first postulate is that there has to be variability. We can see this in the population, right? Can you see variability in this particular trait? Okay, so we know that there's variation. And we know that coat color, the variation that we see in coat color, most likely is due to a genetic basis. But I'm going to tell you this: coat color is not one of those traits that's encoded by a single gene. It's a complex trait that's encoded by multiple genes. So it's a little bit harder to elucidate the genetics or the genetic basis in this case of coat color. But it's there. Okay. The other one is heritability. So basically, in one of her papers that she published in the journal Evolution, one of Hoekstra's graduate students, and probably a series of people were able to show that coat color in mice is a heritable trait. That if you crossed individuals with lightly colored coats with other individuals that with lightly colored coats, that you get individuals with lightly colored coats. Okay? You can keep repeating that over generations. If you crossed individuals with darkly colored coats, you ended up with individuals with darkly colored coats. So that shows that most likely that trait in this case has a genetic basis, and it's also heritable. But this is the most important part of the experiment that we did. So basically, what Hopi's graduate student did was, instead of studying mice in their natural habitat, like live mice that are just found on these barrier islands or these sandy dunes or these old field plantations, what she wanted to do was manipulate, sort of in a paired way, sandy dune habitats and dark habitats, and then determine whether there was higher predation rate. Remember, I mentioned before that there's going to be an environmental determinant of natural selection. Okay? That environmental determinant in this case being tested was whether there was higher or greater predation on mice that were not well camouflaged in a particular environment. Okay, so we had this observation that when you have white sand, you have lightly colored mice. We had this observation that when you have dark soil, you have darkly colored mice. So then the question became, what's causing that significant change? And Hopi had hypothesized that it was likely due to predation rates. So what they did was set out to do an experiment in a number of sites to look at predation rates. These are not actual mice, but they're little Pleistocene models. So they're basically plastic models of mice. Beautiful, beautiful experiment, trust me. Okay. Basically what they did was put plastic models out on the white sand dunes and look at the predation rates of plastic models with very light coat color, plastic models with a very dark color. Plastic models, in this case, with a dark and with a light. Okay? So this is the sandy dune. This is the abandoned agriculture, the old field sites. Interestingly enough, so when you put individuals that are light and dark on the sandy dune, you have much higher predation rates of individuals of Pleistocene models, I should say plastic individuals attempted, okay, higher predation rates on individuals that had a darker coat color because they were not well camouflaged, right? Individuals that had a lighter coat color was better camouflaged. They were not visible to predators. And they quantified some other things. They could actually, based on the marks and on the potential bites, they could actually quantify too the type of predator, or get it to a general group, the type of predator that was causing these predation rates. Experimentally at the same time though, when they placed these little Pleistocene models, okay, on the abandoned agriculture, the old field, what they found in essence was that the lightly colored mice, who were less camouflaged, less well camouflaged, had a higher predation rate. All right. So what they're illustrating here, in terms, is that in this case, competition was non-random. So there were non-random differences that more likely would increase the fitness of mice, okay? When you have lightly colored sand environments, it's going to increase the survivorship and reproduction of light mouse. When you have darkly colored sand environments, or darkly colored soil environments, it's going to increase the survivorship and reproduction of darkly colored mouse, just because of this environmental determinant predation. Okay, so what have we shown is that there is variation among individuals. We can look at this and see that there's a general gradation in traits. It's a complex trait encoded, in this case, by multiple genes. She was also then, in separate experiments, able to show that the variation that we see of this particular trait is inherited. When you cross dark individuals or light individuals, you get individuals that have a similar phenotype. That you will have more individuals then that are born that will survive to reproduce. She didn't necessarily test this. She didn't test whether there was a limitation for resources. But what she did test, in essence, was that some variants survive and reproduce at higher rates than others. She was able then to use these Pleistocene or these plastic models to show that there are higher predation rates on individuals with traits that do not lend themselves to camouflage. Individuals in this case with adaptive traits that were more camouflaged are more likely to have a higher frequency, in this case, of reproduction and survival in that population. And therefore, okay, dark color is adaptive in old field plantations. Light color is adaptive in sandy soils, okay? So that means, and adaptive traits are relative. So it's relative to where you are in terms of geographic location. All right, I'm going to introduce one other experiment. How am I doing on time? 1:16, I'm reading my email? No, I'm just teasing. So that got you to laugh. You laughed right, anyway. It's not really all that exciting. You can read my email if you really want to. My email's never exciting. It's not like something I ever hide from anyone. It's funny. It's like, anyway, I'm going to talk about another experiment now that I have a few more minutes.
Do you want me to remind you of the budget?
Sure, I just wanted to know what time, because you know how much I like to talk. So I can keep you guys. Do you guys want to stay until 5? We just talk about, no? No, okay, no. I was only asking you the time because I know now that I'll only have time to, talk about one experiment, and then I'll wrap up by giving you a couple questions. Okay, another example of an experiment that was done to look at evolution by natural selection was also done on plant populations, and these are shown up here. So you really have, in essence, one particular species, these are known as snapdragons, okay? One particular species that had 2 different color morphs. One white, mostly white, okay, and one all yellow. In essence, generally you find a higher frequency of individuals in a population that are white and a lower frequency of individuals in a population that are all yellow. So you basically have 2 color morphs in the population. Nice, as opposed to studying coat color in beach mice, here we're actually studying a trait that is encoded by a single gene, okay? So there's only one gene. That means there are only 2 alleles in the population too, which is really nice. Makes Mendelian genetics very simple and much easier to understand in this context. Something else you should know. So, because most of you, in essence, either your physiology concentration, there are very few of you probably that are, how many of you are microbiology concentration? Few of you, very good. How many of you are people that would consider yourself plant people? Raise your hand. We're outnumbered, is what I'm trying to tell you. Okay. So I'm going to go back and review some information from 1120. You probably all wanted to forget some of this information from 1120, but I'm going to make you learn it. So have male and female parts, just like animals have, for the most part. Some of them are pathogenic, some of them asexual, okay? Plants, all right, disperse the male reproductive parts through pollen, right? So we define reproductive success sometimes in terms of transfer of pollen. So transfer of these male gametophytes. Okay, so you need a male gametophyte to be transferred to actually fertilize and form a seed in a female plant. We measure fitness. So instead of looking at human populations where it's like the number of babies born, we also have babies in plants. The babies are called seeds, right? Seeds are the same thing. They're the product of fertilization. They result from the union of a male gamete and a female gamete, okay, that fertilize to form potential seed, a little embryo that grows out, a little plant that germinates. So we measure sometimes fitness in two ways when we look at plant populations. We look at the amount of pollen, the male gametes that are moved around, and we look at the number of seeds that are produced in general. Okay. Something else then you should know about this one plant population. As I mentioned before, there is a higher frequency, in this case, of white flowers and a lower frequency of yellow flowers. These are considered what is called a nectar guide. So basically, these are all pollinated. So the way that we transfer the male gametes, the male gametophytes, is through other insects. Generally, plants reward insects or other pollinators with nectar, which is with a carbohydrate or a sugar source. So these are considered nectar guides. So the way to get a pollinator to move pollen from one plant is to offer it a specific reward. So in this case, these are nectar guides, and these nectar guides are signaling to a pollinator, hey, there's a significant source of carbohydrate or sugar here. Okay. Now these, they can see them on the white flowers, but they can't see them as well on the yellow flowers. So these are thought to serve as better nectar guides, and therefore the white flowers, in this case, should have higher reproductive success. Higher pollen means a higher number of seeds, right? Because you need the pollen for fertilization. This is a slide just showing that Mendelian inheritance operates in this population. That you have a single gene that encodes, in this case, for color morphology. 75%, so you have a single gene, a portion of them, any of the white flowers are either heterozygous or homozygous dominant, in essence. So white flowers here, any that encode for the yellow morph, it's due to the fact that they have heterozygous genes. That they have non-functional protein complexes of this particular gene. Okay, so it's non-functional, homozygous recessive. What they did was to conduct an experiment. First, what they wanted to do was see, first look at the number of visits that were made by pollinators in terms of pollination success, and then they also wanted to quantify the number of seeds, or basically, essentially quantify the number of babies. Looking at white morphs and then looking at yellow morphs, okay? Now, what they were able to show is that individuals that had these relatively larger nectar guides had also higher number of bee visits per flower. So they had a higher or a greater success in terms of pollination, or greater success in terms of transfer of that male gamete, as compared to the yellow flowers, which had a lower number of bee visits. And then when they looked at the seeds, they also were able to show that individuals that were either homozygous dominant or heterozygous, and then had the white colored morph, also had a larger number of seeds as compared to the individuals that were homozygous recessive. So what this should illustrate is that in a consequence of a population, what you should see is if you have a higher reproductive success of individuals with a particular color morph, then you should see more of those individuals in the next generation. And that's what happened. So they took all those seeds and then they grew them up. What did they find? So instead of this population, which they started with, that only had 75% either heterozygous or homozygous dominant, now we had 77%. And we had a drop in the number of individuals with the yellow mark. Okay? And it's because these individuals in this case had higher fitness. Doesn't seem like a lot, but over successive generations, what you should see is an increase in individuals and in the population that have that particular adaptive trait. And if they have a higher pollination rate, they're more likely to have a higher seed set. Okay. How am I doing on time? Did anyone email? Did anyone text me? No. You're doing a good job. Okay. Do you guys want some test questions? What? I said I'm recording you. Sure. Okay. I am most likely to ask you what a vestigial. I'm going to tell you. I'm going to ask you what a vestigial structure is. Okay? These are degenerative structures that no longer serve any function. I am most likely going to provide an example of a vestigial structure, one that I have already provided in class. So either it's going to be a hind limb of a whale, or reduced hind limbs that are found in snakes. Okay, snakes have actually lost their hind limbs from their lizard-like ancestors. Or I'm going to introduce the idea of a coccyx. So any of those three, so tailbone. Tailbones are only found in what organisms? Great apes. Okay, great, good. So I will ask you that particular question for the purposes of the exam. Let me go back to chapter 1. Okay, I usually ask, so I'm going to ask you about HIV in the population, and I'm going to ask you to outline an area which has the highest level of HIV infection in the world, and what are you going to respond to? It's going to be one country in Africa, Sub-Saharan Africa, okay? So I'm certainly going to ask you about that. I'm going to probably ask you more questions that I make sure too that you understand this. I'm not going to outline what question I'm going to ask specifically. Make sure you understand the evolution of HIV. By the mechanism of natural selection as a result of AZT. Okay. I'm going to ask about this. I'm going to ask you about the origin of HIV in the human population. The origin of HIV is complex, right? Where did it come from? And so there's HIV-1 and there's HIV-2. And even within HIV-1 you have multiple groups. Right? So, in this case, HIV-1 is most similar to a chimpanzee. But, you can also, groups O and P, we evolved them, we've gotten HIV directly from gorillas. Okay? So chimpanzees are not the only source. It's evolved independently. Separate times. One jump from chimpanzees. We've also had a second jump in this case from gorillas. so it's evolved multiple times in the population. Didn't come from one chimpanzee, didn't even come from one species, came from separate incidences of multiple species. How did it evolve? Or how is it likely that we evolved HIV from SIV in two different ancestral populations, one being chimps and one being gorillas, right? But the SIV, how did we contract the SIV most likely? What? Yeah, but from SIV? Yeah, so yeah, most likely it came from contaminated meat. Like the way that we got it from gorillas or chimpanzees, not how we get it from other humans. Okay? But most likely came from contaminated meat. So either eating infected meat or blood from contaminated meat, killing that potentially. Okay. Now, those are the 3 questions I'm going to give you for the purposes of the exam. And I'm going to review a little bit more once I finish chapter 3 on Monday. And then I'm going to see you guys, bright, shiny, cheery faces, next week.
We're going to ask about historical questions, right? Like who?
Yes, those are only 3 of the questions I gave you. Yeah. Historical questions. I'm sure that you can anticipate.
Right.
I would know who Charles Darwin was. I would know who Thomas Malthus was. I would know who Lamarck was because he presented the first theory of evolution.